In this video, we want to compile the solution set for Biochemistry 202 Imo State University, the year of 2018-2019. The first question says, describe the distinctive molecular features of the major and minor proteins of the muscle. So for us to answer that, you get your writing materials. For us to get that, let's look at the major proteins first. For the major proteins, the first one is the monomeric actin, what we call G-actin. It comprises of 25% of the total muscle protein by weight. Under physiological conditions, it polymerizes to form helical structure or filament, referred to as the F-actin, which means filamentous actin. The second one is the Myosin. It has a double intertwined helical fibrous portion, each with a globular head attached at one end. Myosin of skeletal and cardiac muscles exhibit ATP hydrolyzing property, what we call ATPase activity. And binds readily to the F-actin muscle under some appropriate conditions. Now let's go to minor proteins. For minor proteins, the first one we will look at is the tropomyosin. It is fibrous, made of two helical chains, that's the alpha and beta. They are normally attached to the groove of the F-actin molecule. The second one is the troponin. The troponin system is made up of three distinct proteins, coordinated as TPT, TPL, and TPC. It's unique to all striated muscles. TPT specifically binds to tropomyosin helix as well as other two components of the troponin system. The TPL, that is the TPL, and the TPC. The TPC is a calcium binding protein. The next question says, name any three synthetic nitrogenous bases and their therapeutic usefulness. The first one we'll talk about is the azatopane. So this azatopane is useful in organ transplantation as suppressor of events involved in immunologic rejection of tissues or organs. The second one is allopurinol. It is useful in treatment of gout and certain types of kidney stones. The third one is fluoracil. Is used for treatment of cancer. The next question says, explain the following terms: nucleoside, base pair rule, phosphodiester. So let's begin with the first one, nucleotide. This is a phosphate ester of nucleoside. Naturally occurring nucleosides can also exist as nucleoside monodiphosphate. This is the structure of it. Then comes down here. So this is adenosine 5 monophosphate. That's AMP. Remember, there's a double bond here and here and also here. So the structure is complete. The second one is nucleoside. It's a compound that have a purine or pyrimidine nucleobase covalently bonded to the D-ribose or 2-deoxyribose sugar through an nitrogen beta glucosidic linkage. The bond linking the two moieties the nucleobases and the ribose or deoxyribose sugar is through the N1 and C1 atoms respectively. An example of that is the uridine. We have O double bond, then and here is double bond, N is here, N is here, the N here has this, and then we have, so there we have O. Okay, so here we have so here is the C1. So the next one is the base pair rule. Now, this law or this rule states that DNA base pairs are always adenine with thymine. That is A to T. And cytosine with guanine. Cytosine with guanine is C to G. A purine always pairs with a pyrimidine and vice versa. And this rule is also known as Chargaff's rule. Chargaff's rule. The next one is phosphodiester bond or linkage. This bond occurs when exactly two of hydroxy group in phosphoric acid reacts with hydroxy groups on other molecules to form two ester bonds. The bond involves this linkage, that's COPO2 minus OC. The next one is mutations. It refers to a change that is induced by chemical or radiation or biological agents, which is mutage, in one or more purine or pyrimidine base, thereby altering the nucleotide sequence of a gene, endangering a defective gene product, that's protein. Some consequences of gene mutation could be sickle cell anemia and so on. So that engendering defective is what causes the alteration. The next one is mutations. This refers to a change that is induced by chemical or radiation or biological agents, which we call mutage, in one or more purine or pyrimidine base, which alters the nucleotide sequence of a gene, engendering a defective gene product. Some of the consequences of gene mutation are sickle cell anemia and so on. The next question says, describe the basic pathways of 1. Lipogenesis, 2. Lipolysis. Let's begin with the first one, lipogenesis. Lipogenesis involves the esterification of free fatty acid with a trihydric alcohol activated in the form of acetyl-CoA and glycerol 3-phosphate respectively. Now the pathway goes this way. O double bond C OH. Here we'll have, I don't know whether it will enter here, CH2 and then CH3. Okay, it has entered. Then we have an arrow. Here we have CH3, CH2N, and then C. Then we have CoA. Then that is activated fatty acid. Then this is acetyl-CoA, activated fatty acid. Then we have CH2OH, CHOH, CH2OH. Then from this we have CH2OH, CHOH, CH2OH. That now goes to POO. So here is glycerol propene 1, 2, 3 triol activation. Then here is glycerol 3 phosphate activation. That here is now activated glycerol. The second one is lipolysis. It is a term used to connote the hydrolysis of lipids, such as triacylglycerol. The end products of the hydrolysis of triacylglycerol are acidic. They are acid, so that's the end product. So here is the pathway. You can pause the video to take a closer look at it and study it. The next question says, define the following terms. The first one is ketone bodies. They are substances produced by the liver during gluconeogenesis. Acetyl-CoA is converted in the liver into acetoacetate, which can be further reduced to form beta-hydroxybutyrate. The next one is metabolism. It is the sum total of all the chemical reactions within a biological system related to the management of the supply of energy to power cellular activity and the generation of molecules for cellular synthesis. The third one is catabolic pathways. This process degrades complex compounds into simpler molecules, releasing building materials and chemical energy. They are exergonic, that's energy releasing. We can have, like for A now, we have all these particles and it moves. So it releases energy. It can be Xi1. So the second one still moves, releases energy. We call this enzyme 2. Still moves forward. We call it enzyme 3. Then is C, D, move releases energy. So we call this E. So the other one is the word exergonic. Exergonic energy releasing. So the other one is anabolic pathways. This process leads to the biosynthesis of complex molecules from simpler components. So since molecular construction requires energy, anabolic reactions are said to be endogenic. Endogenic, that's energy absorbing. This is energy releasing, energy absorbing. The other one is glycogenolysis. It means breaking down of glycogen. In glycogenolysis, glycogen stored in muscles is converted first to glucose 1-phosphate and then into glucose 6-phosphate. Two hormones which control glycogenolysis are peptide and glycogen. Then the other one is glycogenesis. It's simply the formation of glycogen from glucose. In synthesis of glycogen, 1 ATP is required per glucose, incorporated into the polymeric branch structure of glycogen. So the next question says, represent the glycolytic pathway in a step-by-step pathway and describe the 5 major important facts about glycolysis. So this is actually the pathway. This is the pathway. You can pause the video and study it. So this is actually the pathway. So the important facts about glycolysis is that glucose produces 2 pyruvic acid molecules. The next one is that ATP is initially required. The third one is ATP is produced. And the next one is if the cell is operating under aerobic condition, that is oxygen is involved, then NADH must be reoxidized to NAD plus by the electron chain. The next question says, discuss the biochemical rules of 5 micro minerals, their dietary sources, and deficiency symptoms. The first one we'll look at is iron. It is part of a molecule, that's a hemoglobin, found in red blood cells that carried oxygen. It carries oxygen in the body, needed for energy metabolism. The dietary sources, that's organ meat, red meat, fish, poultry, dried fruits, egg yolks, etc. The deficiency symptoms is anemia, slow social cognitive development in children, feeling weak and tired. The next one we'll look at is zinc. It has a function in taste perception, wound healing, normal fetal development, production of sperm, normal growth of organs and sexual maturation, immune system, and also health generally. The sources are, the dietary sources, they are meat, fish, poultry, vegetables. The deficiency symptoms, loss of appetite, taste or smell, decreased function of the immune system. Another one is iodine. It helps regulating growth. The sources, seafood, iodized salt, bread, dairy products. The deficiency symptoms can result in enlarged thyroid gland, that's goiter, in the neck. The next one is fluoride. It is involved in the formation of bones and teeth, help in prevention of tooth decay. Sources, drinking water, fish, and most teas. Deficiency symptoms, tooth and bone weaknesses, that is tooth decay. Then we also have selenium. We have selenium. It's an antioxidant. The sources is their meat, seafood, grains, and so on. Deficiency symptoms, weak immune system, tumor formation, heart and liver disease. Another one is copper. It helps to form healthy bones and joints and nerves, essential in the formation of collagen. Then the deficiency symptoms, what we call Wilson disease, Menkes disease, and so on. The next question says, describe the role of vitamins listed below in biochemical reactions. Thiamine, riboflavin, nicotinamide, ascorbic acid, biotin. Then let's begin with the first one, which is thiamine. Its role is to serve as coenzyme in the decarboxylation of alpha keto acids, and keto sugars, and is therefore important in carbohydrate metabolism. The next one is riboflavin. It acts in the body in the form of the coenzymes flavin mononucleotide, that's FMN, flavin mononucleotides, and flavin adenine dinucleotide, which is FAD. The next one is nicotinamide. Humans convert niacin to nicotinamide, which contains a pyridine ring and is required for the synthesis of the coenzymes NAD plus and NADP, which are used in dozens of oxidation reactions. The next one is ascorbic acid. It functions as an antioxidant and as a coenzyme utilized in oxygenation reactions. Ascorbic acts as a reducing agent, maintaining the ion atom of enzymes in the ferrous, that's Fe2 plus, rather than ferric, Fe3 plus state. It also improves intestinal absorption of iron. The last one is biotin. Biotin. Its essential role is to carry activated carbon dioxide